Hello students, we are continuing our topic cell cycle, and what are the changes which is happening to the cell in the interphase and in the prophase 1 and 2 that we have already discussed. And if you haven't watched it, I will give the link in the description. You can go through that. Okay. So now we are at the second phase of karyokinesis, otherwise metaphase. The first phase was prophase. In prophase itself, the spindle fibers started to capture the chromosomes that we have already seen. But in prophase, the chromosomes were not in the equatorial plane. Means all these chromosomes were not in the same plane. It was not aligned like this. Okay. All these chromosomes were the different levels of the cell. Okay. So the major event or the major change that you can see in the metaphase is that all the chromosomes will be in the equatorial plane of the cell. If you are getting a picture like this to identify the stage of the mitosis, without any doubt you can write this is the metaphase, because you can see that all the chromosomes are in the central plane of the cell. Okay. So how this arrangement is being possible? See, in metaphase you can see that the spindle fibers are attached to the kinetochore. Kinetochore means there is a special region on the centromere where on which the spindle fibers are attaching. Okay, the region of the centromere where the spindle fibers are attaching to the chromosome. That region is known as the kinetochore, and the spindle fiber will get attached to the kinetochore. Okay, like this it will get attached to the kinetochore, and this is coming from where? It is coming from the centriole, from this pole. Okay, this centromere will be, and from this pole this spindle fiber will be. Okay, so what is really happening means this spindle fiber will start to pull the chromosome to this side, and this spindle fiber will start to pull the chromosome to this side. Okay, this is really happening, and it will create a pressure on the centromere, and finally it is going to be split. That is in the next stage. Okay, so what are the changes which is happening in metaphase? Means two changes that you have to write. That is, the chromosomal fibers will get attached to the kinetochore, and because of the contraction of the spindle fibers, all these chromosomes are going to be aligned in the equatorial plane of the cell. These are the two observable features of metaphase. Okay, and see, there are two types of spindle fibers that you can see over here. Okay, see, look at this spindle fiber. It is not connected with any chromosome. It is a continuous spindle fiber which is starting from this pole and it ends in this pole. Okay, in between two poles without attached to a chromosome, a spindle fiber is here, and this kind of spindle fiber is known as a continuous fiber. Otherwise you can call it as a interpolar fiber. Okay, and another type of spindle fiber which is discontinued because of the attachment of the chromosome. Okay, so it is started from this pole and it is going, and meanwhile it is get attached to a chromosome, so that it is not a continuous one. So that we can call this spindle fiber as a discontinuous spindle fiber. Otherwise, it can be called as a chromosomal spindle fiber, so that it is attached to a chromosome. Okay, there are two types of spindle fibers are there: chromosomal spindle fiber, otherwise the interpolar fiber. Okay, so this is what I have told you, that the spindle fiber is attached to the kinetochore, and this spindle fiber will start to pull towards this pole, and this will start to pull towards this pole, and it is getting it is creating a pressure on the centromere. Finally, the centromere will get split. That is really happening in the next phase. The next phase, otherwise anaphase. Okay, in anaphase, what is really happening means the splitting of the centromere. See, when this chromosome is getting split, it will be like this. Okay, you can see that this is one half of the chromosome. One half of the chromosome is here, and another half will be here. What is really happened? This centromere has splitted. Okay, like that it will get split. That is the major feature of anaphase. In anaphase, the splitting of the centromere will be there. Okay, that is the first feature or the major feature in anaphase. And next feature is that the sister chromatids separates and move to opposite poles. So the sister chromatids, because of the splitting of the centromere, the sister chromatid will start to move to the opposite pole. This is a sister, this chromatid, this sister chromatid is moving to this pole and it is going to move to the opposite pole. Like that, the sister chromatids will get separate. So when the centromere is splitting, you can see that another kind of fiber will be there in between. Okay, because of the splitting of the centromere, one another kind of fiber will get created, and that fiber is known as the interzonal fiber. Okay, so what is interzonal fiber? The fiber which is created because of the splitting of the centromere, that is known as the interzonal fiber. This is the first stage of anaphase. Okay, in first stage of anaphase, you can see that the centromere is already splitted and the interzonal fibers are started to form and the sister chromatids are started to move to the opposite pole. Okay, it is not in a big distance. Okay, it is not that much huge distance is not there in between. Still, it started to move to the pole. A small distance that you can see in between the sister chromatids over here. This is the first anaphase. Okay, and in late anaphase, really what is happening means the sister chromatids will far move to the pole. Okay, it will more move to the pole and you can see a big distance in between the sister chromatids. Otherwise, the interzonal fibers will be large than that of the early anaphase. Okay, so and in late anaphase, the sister chromatids will be more close to the pole. Okay, it will be more close to the pole, and in an animal cell you can see that the furrows will start to form here. Okay, the furrows will start to form here. It is the starting of the cytokinesis. Okay, the furrows will start to form over here in the animal cell. So what are the major features in anaphase? The centromere will split, sister chromatids will separate and it will start to move to the opposite poles, and furrows that you can observe in the cell membrane, okay, of an animal cell. These are the major features of anaphase. And the next phase, that of the karyokinesis, that we are going to discuss in the next class. Thank you.